eyes. So they never develop any evidence of an eye structure, really, in this mouse. In addition to that, though, they have some other things that go wrong. For example, look at the nice nose with nostrils and mouth and chin in this wild-type mouse. And over here in the mutant mouse, those structures are malformed for sure. So they're not quite missing, but they're not quite normal either. So now we're seeing PAC6 not only needed for eye development, but for some other stuff too, including nose development and craniofacial structures. So in the embryo, PAC6 is expressed in the diencephalon. That's about where you'd expect it to see. But in the middle, between this, we're going to see a region where sonic hedgehog, a paracrine factor, is expressed. So in the wild-type situation, we should get sonic hedgehog in the middle and then two separate PAC6 domains. And from the PAC6 domains, now we get eyes developing. And so what can happen if you have a sonic hedgehog mutant? So we're not really specifying the middle territory that should separate these. Now the two fields that become eyes move closer together. So these are two arrows here pointing to those optic fields, or even might get fused into a single entity. And then this may give rise to the cyclopia phenotype down the line, where the two eyes are either moved more towards center and closer together, or fused into one. And all of this is subject to evolution. Could play with this scenario and perhaps adjust things as needed for that particular species. So here is a situation where we have some fish that are either in the surface dwelling population or in a very dark, deep underwater cave dwelling population where there is no light and they may not actually need any eyes down there. And what was identified in this population is a variation in the way sonic hedgehog was expressed. So sonic hedgehog is actually kind of overexpressed in this cave dwelling population to the point that now there's no region where PAC6 could be expressed, and then those fish don't develop any eyes, which seems to be something that maybe could be advantageous to them in their population where they don't really need the eyes in any way. So here is an indication of PAC6 within a hierarchy of genes. It is a transcription factor. It's regulating some of them downstream, but then also there are things upstream that regulate PAC6. And in general, the theme that we're seeing here is that you need to have PAC6 in order to get eyes to develop. So PAC6 is necessary for eye development. That's a key point. Upstream of PAC6, we have this gene called RX1 that's also expressed in the embryo, in the neurula at about this stage, and it's regulating the expression of PAC6. So if you have a mutation in this RX gene, then now you're going to get phenotypes related to eye development, where generally there would be reduced eyes or no eyes, and that is because of RX1 regulating PAC6. So that's always a possibility too. And here is the homozygous RX knockout in mice, and they lack the eyes. So that's probably because of the way that RX is regulating PAC6. So if we don't get PAC6, now we're not going to get any eyes in mice as well. So now PAC6 could still be expressed differently in different tissues, such as something needed for survival. But in this particular case, if we can knock out PAC6 in the eyes, at least we don't get any eye development. And in human, there actually is an RX mutant phenotype out there, and it's a developmental disorder where they don't develop ocular tissue. So it's kind of interesting to see that these same pathways seem to be in play in human embryos as well. Okay, so there are lots of different eyes out there in the animal kingdom. The sockle eyes are functionally, structurally different than human eyes, and yet both of these seem to be using some of the same genes, like PAC6, and employing these in the diencephalon early in the embryo start this process of eye tissue differentiating. And so one biologist at the time, Walter Gehring, who was in Switzerland, he's like, you know, in Drosophila we have this magnificent ability to manipulate genes. And why don't we do the dream experiment, which would be to express the PAC6 in places in the Drosophila embryo that are not eyes. So it's basically a move-it experiment. He wants to now express PAC6 and see, are we going to get eyes in places that normally we would not? And so in this case, if he expressed PAC6 ectopically, that means in a different time or place, now he would get eyes developing at various non-eye places in the embryo. So you could see eyes coming out of the antenna. You could see eyes on the wings, even eyes on the legs. And because eyes have this really recognizable feature of pigment and structure and organization, you know, it was easy to verify that this in fact was eye organ tissue developing in these different places. And so now keep in mind, this is one gene, a single gene. You're just expressing it at the wrong time and place, and you are initiating an entire program of organ development from that one gene. And wow, this really made a huge headline because it showed that PAC6 is sufficient for eye development. And you know, it varied how far along these eyes could get, which may mean that there are different competencies here, but just the possibility to do this was kind of mind-boggling. Who would ever think that one gene could do such a thing? Here's just a picture of some of these through electron microscopy. And this gave rise to the kind of a term, an idea, that we might call PAC6 a master regulatory gene. It's like the godfather gene of eye development. You know, this guy, you put him out here, he organizes everything. You know, he sends out the minions and we get the job done. We have an eye on our wing or our antenna. So, you know, people were really excited about this and kind of wrestled with the idea of, could there be such a gene? And would it work in animals as well? And in fact, actually I should tell you, you could even take the human PAC6 gene and express this in Drosophila, and that it will work also. So even your human version of the gene could work in Drosophila to establish these ectopic eyes. So now here's a situation where we're kind of mixing and matching which tissues are PAC6 positive and which are PAC6 negative, and that's testing where do you actually functionally need PAC6 in order to get eye development. Would it be in the ectoderm, which is overlying the outside of the eye? Would it be in the optic vesicles? Either one or both. And so in this situation, one or the other or both is mutant here. And when both are mutants, you don't get anything, which is our control situation down here. But as long as PAC6 is wild type in the surface ectoderm, now you're going to get lens induction occurring. So it turns out you actually need PAC6 in the ectoderm rather than in the vesicle itself, which was maybe a little bit surprising. But just to show you an experiment here that taught us about where the PAC6 is needed in human eye development. You knew it had to happen. Somebody wanted to do that move-it experiment in a vertebrate as well. So here they are expressing mammalian PAC6 frog PAC6 in an ectopic fashion. And in this case, it will generate some ectopic eyes. So these are these little black pigmented structures here indicated by the arrows. All of these are ectopic eyes. If you section through them, you can recognize some of the tissues, such as lens tissue. These are ganglial cells, interneuron cells, photoreceptor cells. But you know the organization is not great. So it's not really a perfect ectopic eye, and I don't think they could actually see, or maybe not even perceive light. They definitely don't have a neural connection going back to the brain, so functionally they're not eyes, but morphologically, in terms of cell base, they are on the right road. So there's quite a bit of power that this master regulatory gene has, even in a mammalian situation.
With Drosophila eyes, perfect ectopic eyes, or
The Drosophila eyes are usually not perfect ectopic eyes, but they are compound eyes. So they're these compositions of single eyes, and many of the sections through the single eyes look really good. So the way that all those cells are organized is totally fine.
okay.
And so those also probably don't have the neural connections to be functional, but structurally they look really good.
Okay.
Okay, so here is just looking at some of these ectopic eyes in the frogs with markers to, again, try to convince ourselves that these cells really are differentiating in the way that we think they are, and they're taking on the various different cell fates that you can find within eyes. So they do get pretty far. Okay, so this brings us to this idea, master regulatory genes. Here is Russell Crowe in Master and Commander, so I thought he'd be a good one to throw up here while we talk about this. And to what extent is this concept of a master regulatory gene a useful concept? And at first, the idea went out and people were excited that, yeah, what if we could find genes that would have this sort of tremendous organizational power within the embryo? And PAX6 is as good an example as you could probably get. But on the other hand, there's some limitations to this as well. So for one, PAX6 is a master regulatory gene for eye development, but that's not the only thing it does, right? It was involved in nose development too. It might be involved in some early function that's needed for survival, because if we knock it out, we don't get survival, as in every case. So PAX6 is not just eye development. So that kind of muddies the idea of one gene being a master regulatory gene. Another thing is, if you express PAX6 ectopically, not every single cell becomes an eye. You get these structures that form eyes, but not every cell is competent to respond to an inductive signal by PAX6. So that's another mitigating factor to eye development, because you need something beyond PAX6, even though PAX6 itself goes a certain distance in being sufficient for eye development. Then another thought here, PAX6 is not the only gene in this whole process. It's a transcription factor and it's part of a hierarchy, but there are other genes that are needed to fulfill the dream of eye development. And so here is iris, and one of the regulators of iris in Drosophila is another gene called twin of iris. If you ectopically express twin of iris, you can get the same thing, these ectopic eyes. So, we happen to fasten onto iris, discover its properties first, but it's not the only gene that really has this ability. And yet, the reason here that twin of iris has ability is because it's regulating iris. So it does kind of come back to iris, but it's kind of an oversimplification to think about one gene as being the master regulatory gene that could really do the entire job. So, and then there are other downstream genes, and many of these are the same in humans and Drosophila. And sometimes you'll see that humans have multiple copies of some of the genes, or Drosophila has multiple copies. So that also kind of contributes to muddying this vision of a single master regulatory gene. So, one of the study guide questions is kind of asking, what were some of the reasons that are for or against the concept of master regulatory genes? And I think today that it's a little bit passé to say master regulatory genes are truly in charge, and yet the concept of it still sort of remains. And you can find particular interesting genes like this one that do have some really tremendous abilities in the way that they influence cell fate and organ development. Any thoughts or questions on this? Yeah?
So does PAX6 considered a Hox gene that has the homeobox in it?
PAX6 would qualify to be a homeobox gene because it does have the homeobox conserved domain. It's not actually called a Hox gene because that's just a name of some of the genes that were in those arrays that were duplicated. So its gene map location is elsewhere within the genome, and it happens to have a different name. Okay.